Hey everyone, this is Elad from Astrolab Diagnostics, and today I would like to talk about different perspectives in differential expression analysis. In other words, how to take a given experiment design and inspect it from different viewpoints. Briefly, differential expression analysis, its goal is to identify markers that change an expression across given population. So let's say that we have responders and non-responders. We would like to examine different cell subsets and different markers. So for example, how does the level of PD-1 change between T cells in the responders and in non-responder populations? To demonstrate differential expression analysis, I'm going to use an immune monitoring study from Mount Sinai Hospital. In this case, we're looking at peanut allergic patients. We have 6 patients and 3 healthy donors. We have 3 different conditions for the samples. We have media, which is negative control, anti-IgE, which is positive control, and we have the peanut extract, which is the experimental conditions. We have 2 different time points, and overall we end up with 54 different samples. Now, there's quite a lot of covariates here, right? We have patients versus healthy donors, we have different time points, we have 3 different stimulation conditions. We can shuffle this matrix in many different ways. I'm going to demonstrate a couple of ways using the Astrolab platform. So one way is to just look at the patients. So let's say we would like to look at the patients at the 15-minute time point. I'm going to click on this analysis. In this heat map, every row is a subset, every column is a marker. The deeper the color, the greater the fold change in that marker. And if we look at the basophils at CD61 and CD63, we can scroll down and see that there is a baseline level of CD61 in the media condition. It goes up in the peanut extract, and this is comparable to the anti-IgE, the positive control. Likewise, looking at CD63, we see a similar trend. Here is the baseline for the 6 patients, here is the peanut extract, and here is the anti-IgE. We can see how the peanut extract is very similar to the positive control. Now at this point, we only looked at the patients. We can similarly look at the healthy controls, and when we do that, we generate the same visualizations. Now we'll see that the peanut extract is equivalent to the media, not to the positive control, both for CD61 and CD63. So this was one way to look at the data. We examined the patients as one group, the controls as another group, and we're examining just one time point comparing the 3 stimulation conditions. We can also choose to include the controls and the patients in one visualization. And you'll notice that this heat map is much more populated, it has many more colors, because now we will also identify differences between the controls and the patients as individuals, which might not be relevant for the analysis that we are doing here. We're not interested in heterogeneity within the population. What we care about is heterogeneity in response to stimulation. And looking at the basophils, and again CD61 and CD63, we now have 6 different boxes here. The first 3 are controls: media, peanut extract, and anti-IgE. The latter 3 are the patients. Again, media, peanut extract, and anti-IgE. We can see the same trend as before. We can see how the media is the same between the controls and the patients, and then the peanut extract for the controls looks like the media, while for the patients, the peanut extract looks like the anti-IgE. It's the same data as before, just a slightly different perspective. Beforehand, we focused just on this group, separately from the other group, and now we're looking at both patient populations and comparing the level of 61. The same trend, by the way, is observed for CD63. Once again, this figure mimics the 2 figures we've seen before pretty well. However, we are examining the donors and the patients in one figure. So just to summarize, different ways to look at the data. If you have any more questions about analysis, please reach out. I'm Elad at astrolabdiagnostics.com. And please make sure to subscribe to our LinkedIn and to our YouTube to get more of these videos. Thank you for your time and have a good day.